This is an overview of the macromolecules. So, carbon is the backbone of life. And we call all compounds that contain carbon organic compounds. Why is carbon so darn important? As we talked about earlier, it's because of its valence shell. The fact that it has those 4 open spots makes it amenable to bonding with other atoms, other elements. And its favorite partners in bonding are hydrogen, oxygen, and nitrogen. The most important chemical groups help with functionality, and that's because of their reactivity. So, in particular, these functional groups are the hydroxyl group, carbonyl group, carboxyl group, the amino group, and the phosphate group. These are the important groups that you should know as a biology student. Again, the location of where these functional groups are on molecules helps to determine their functionality, and so that matters as well. Now, the big polymers that we are interested in are called macromolecules because they're really large. They're made up of monomer subunits. These are simpler, smaller molecules that are joined together in specific ways with specific types of bonds. All polymers are made through a dehydration reaction, and they're all broken down through a hydrolysis reaction. There are 4 different kinds of macromolecules. They are carbohydrates, they are lipids, they are proteins, and nucleic acids. First, an overview of the carbohydrates. Carbohydrates contain carbon, hydrogen, and oxygen, and they function as either an energy fuel molecule or a structural molecule. There are different kinds based upon their size. The simplest, which only contain a single sugar, are called monosaccharides. 2 monosaccharides joined together form a disaccharide, and that is known as a glycosidic bond that holds those 2 monomers together. Polysaccharides contain many sugars, and there are several types of polysaccharides. There are glycogen, starch, cellulose, chitin, and peptidoglycan. Of particular interest should be the difference between starch and cellulose. They look pretty much the same, but there is one small but important difference, and that difference is in the glycosidic linkage. Specifically, cellulose has a beta 1:4 glycosidic linkage. The glucose are flipped in their orientation, and that creates a different type of a bond. Starch, on the other hand, has an alpha glycosidic linkage, and all of the glucose molecules are in the same orientation. So what we can see here is a small difference in structure will lead to a difference in function. So you should ask yourself, what does starch do versus what does cellulose do? 2 different things because they have 2 different structures. Next are the lipids. And the lipids are a very diverse group. There are 3 different kinds of lipids. They are the fatty acids. Together, when you have 3 fatty acids, it's called a triglyceride. There are the phospholipids, which are found in membranes, and the steroids, which are another interesting or diverse group. But what do all 3 of these have in common? They all have what's known as a hydrophobic region. Again, recall, hydrophobic means they don't like water. They specifically shun away from the polar water molecule. If we take a look at the triglycerides, these 3 fatty acid tails are held together by an ester bond. If you look at the phospholipids, we see that it's 2 fatty acids, and these are the tail, and the head is a phosphate group, which is hydrophilic. The tails, though, are hydrophobic. And then finally, steroids have an interesting 4-ring structure with carbon as the basis. Again, the steroids are hydrophobic. Proteins are the next type of macromolecule, and they are really diverse. There are a lot of different types of proteins out there, and they perform all kinds of different functions. But if we take a look at protein structure, the monomers that make up proteins, also known as polypeptides, are amino acids. There are 20 different amino acids in nature, and each of them has the same structure, except they all have a unique R group, and that R group gives them unique chemical properties. The R group is also known as the side chain. These 20 amino acids can then further be divided into 3 different subgroups, again, based on the overall chemical makeup of their R groups. The 3 subgroups of the proteins are the polar group, the nonpolar group, and the electrically charged group. Proteins have a particular structure, and that structure depends solely on the amino acid sequence. If we take a look at protein structure, there is a hierarchy. The first level is known as the primary structure. Again, the amino acid sequence. Starting from the beginning, which is the N-terminus, and going all the way down to the end, known as the carboxyl terminus. The secondary structure, which consists of either alpha helices or beta pleated sheets, are when the primary structure starts to fold and wind in on itself. The tertiary structure consists of 2 or more different types of secondary structures. And then the quaternary structure is when you have multiple subunits coming together and interacting to form the larger protein complex. And again, protein shape determines protein function. So if you change the amino acid sequence, you can potentially alter the function of the protein. And finally, we have nucleic acids. These macromolecules are important because they store, transmit, and express genetic material. The nucleic acids include DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Nucleic acids are made up of the monomers known as nucleotides. These nucleotides are comprised of a nitrogenous base, a sugar, and a phosphate group. These nucleotides are linked together by a phosphodiester bond. Then they can base pair with the other strand of DNA via a hydrogen bond. The important thing to keep in mind about DNA is that it is double-stranded, and again, it is anti-parallel. The double-stranded nature comes from the hydrogen base pairing between the complementary bases. A always base pairs with T, and G always base pairs with C. It's anti-parallel in that one strand is oriented in one direction, from 5' to 3', and the other strand is oriented in the opposite direction, flipped over. For RNA, it's a single-stranded molecule, and the sugar is just ribose. Finally, the other unique thing about RNA is that we have uracil instead of thymine. So there's an U instead of a T. Great. So I hope this helps you to prepare for our lecture on macromolecules a little bit better.